Sorry. Okay, let's go.
Hello, everyone. Good morning from Seattle. Sarah Sweger is going to be talking today about Bayesian probabilistic analysis of deer data. Sarah hails originally from Pennsylvania. She went to undergrad at Duquesne University in Pittsburgh, Pennsylvania, where she looked at studying the effects, simulating the effects of crowding on the binding of photosynthetic protein system with Jeffrey Madura. She came to Seattle to join Sean Stoll's group almost 4 years ago now, and today she's going to be where she's worked on deer processing and deer theory, and today she's going to be sharing some of that work with you. So Sarah, feel free to take it away.
Thank you. Thanks everyone for coming. So as Lizzie said, I'm Sarah Sweger and I work in the Stoll Lab at the University of Washington in Seattle. And I'm going to talk today about some of our recent work applying Bayesian statistical methods to parametric models for deer data. So just brief overview, I want to start by briefly introducing what deer is and why we care about a robust analysis. Our setup of a Gaussian mixture model and implementation of the Bayesian inference methods illustrate the method through several synthetic and experimental examples, and then what we can conclude about this work so far. So starting very big picture, proteins are a central topic of focus across a range of scientific fields because of their crucial roles in so many processes. And specifically, it's the conformational changes within these systems that often dictate their ability to function in processes such as signal transport across a membrane, signal transduction within a cell, catalysis of substrates to products, and even cellular motion. And as we're familiar, there are a lot of experimental methods that allow us to see what's occurring on the protein level, and among those is deer spectroscopy. So double electron-electron resonance, or DEER, extrapolates a full range of distances on the nanometer scale. And this is typically done by placing two unpaired electrons at specific sites of a protein of interest using a method known as site-directed spin labeling. Then when placed within a magnetic field, the magnetic dipolar coupling between these unpaired spins can be measured with the DEER technique, and from that a distance can be determined, generally in the range of 2 to 10 nanometers. So now say that my protein undergoes a conformational change, the distance between my spin labels shifts, that shift would then be reflected in my obtained distance distribution. However, in our sample tube, we likely have a mixture of these conformations, which means our final distribution would look something like this. And so this is a really simplistic example, but it's intended to show one of DEER's key advantages is that it provides not just average distances, but a full probabilistic distance distribution of multiple molecular conformations. So now actually obtaining this distance distribution is a non-trivial task. Moving from this time domain signal, which is what we actually get when we run a DEER experiment, to a distance distribution is an ill-posed problem. And what that means is minor changes in this data can lead to significant changes in the output distance distribution. And there are several approaches for moving between these two domains. The analysis is generally broken up into two fields, utilizing non-parametric or parametric models. Each have their advantages and disadvantages, so I'm not going to get into that in the scope of this talk. But under non-parametric, the primary method, as most of you are probably familiar, is Tikhonov regularization, which is a form of penalized least squares fitting that allows us to capture the misfit between a non-parametric model and the data, as well as penalizing for certain properties of the, certain unwanted properties of the distance distribution. And so now the next natural question would be, what error analysis methods are we using to evaluate any error that could be introduced in these complex data analyses? And so for Tikhonov, it has two primary forms, a statistical approach known as Bayesian inference, and a sensitivity analysis that evaluates the effect of certain regularization parameters on the output. But since this talk is focused on parametric models, that's all I'm going to say about non-parametric, and we're going to switch gears and talk about this primary analysis method, which is Gaussian mixture models. And so this analysis assumes the shape of the distance distribution to be a sum of Gaussians. And to be clear, this is done for mathematical ease and not because we necessarily believe that the distribution is Gaussian in shape. So the primary method of error analysis here is to statistically evaluate the fit parameters using a method known as propagation of error. And so what we've done is take this Bayesian inference method previously applied to Tikhonov regularization in a limited capacity and apply it to our Gaussian mixture models. So now how have we actually set up this probabilistic multi-Gaussian model, beginning with the time domain. As this is a parametric model, we must define a set of parameters theta. Starting with our traditional Deer equation, we have VM, which is our noise-free model time signal, which is a product of three components: the maximum signal amplitude, the signal from the intramolecular coupling, sometimes known as the foreground, and the signal from the intermolecular coupling, which is often referred to as the background. So now when I expand this equation, I see my relevant time domain parameters, which is again this maximum signal amplitude V naught, our modulation amplitude lambda, and our background decay rate K. So now, as most experimental Deer data is generally not noise-free, we apply a Gaussian distributed noise around the model data with a standard deviation of sigma, which we have then incorporated in our parameter set. Expanding this to the distance domain, we're assuming, as I've said, that the distribution is going to take the shape of a sum of Gaussians. So when considering multiple Gaussians, three relevant distance domain parameters come up: the amplitude of each Gaussian, its peak position, and peak width at the full width half max. And these last two parameters are obtained by arbitrarily fitting a Gaussian to this distance distribution. And so this is now our full parameter set for the model, keeping in mind that the last three may be vectors depending on the number of Gaussians in the chosen model. So now implementing this model within a Bayesian framework begins with an understanding of Bayes' theorem. And Bayes' theorem output says that what is the probability of my specific set of parameters being true given my model choice, any data I've collected, and whatever background information I might apply to my analysis. And so for simplicity, we will rewrite these as theta, m, v, and I. And this entire term is what's known as the posterior. And so now this posterior is equivalent to a few other components, a product of a first likelihood function, which says what is the probability of collecting this data for a given parameter set, model choice, and background information, and then aptly named a prior. So what is the probability of my parameter set, just taking into account the model choice and any background information that I might have without considering the data. And then finally, a normalizing constant, just saying what is the probability of this data occurring under any circumstances. So we've defined the model parameters, we have a fundamental understanding of Bayes' theorem, and now we need to set up a prior for each parameter that I've shown. So to give an example of how these priors are chosen, we can look at the modulation depth. So the modulation depth is just the efficacy of the pump pulse. And so what that means is just how many of these pump spins are actually inverted by this pi pulse. And so if this is a percentage, that means the most diffuse knowledge that I could apply to this system is that it must lie somewhere between 0 and 1, and that it has a uniform possibility of all values. But now say that I know a little bit more about my system. Maybe I'm running a nitroxide sample at Q-band with seq-tan pulses, and it's a Wednesday, and the spectrometer likes to run on Wednesdays. So maybe my modulation amplitude is likely going to be in the range of 40 to 60%. So something like a beta distribution that favors higher probabilities in this region would probably be more appropriate. And so both of these would be perfectly allowable priors. And so we apply this similar logic and define appropriate priors for all of the parameters of our system. We take these and the raw Deer data and use Markov chain Monte Carlo methods to generate thousands of samples of these priors, of which the parameter values are then used to calculate a likelihood function, which measures the misfit between the model and the data. And then over the course of these thousands of samples, the priors are refined, and the direct output of this Bayesian method are these posterior distributions. And so this is just an exemplary case where we're starting with these fairly diffuse priors, and the method is quite confident that it is able to narrow the posterior distributions significantly. So now to actually show the model through various illustrations, we'll start with synthetic examples. And for these synthetic examples, we utilized the T4 lysozyme test set generated by Thomas Edwards and Stefan in 2018. And we started with a simple distribution that looks relatively Gaussian in shape. And so then from this distance domain, we generated two time domain signals, one labeled V good and another V poor, where V good has relatively favorable values for the modulation depth, background decay rate, trace length, and noise level. And then V poor has comparatively less than favorable values for all of these parameters, which is quite obvious from looking at them. And so this gives us a sort of ideal case and then a more edge case with a higher uncertainty to approach the method. So first we'll look at the direct output of the method, which is the posteriors. And so these dashed lines for R naught and W, as I've said, are the values that we got from directly fitting a Gaussian to that synthetic distribution. And then for the other parameters, the black dashed lines indicate the values that were used in generating the signal. And so these are what we're taking as our ground truth. And so for this longer, less noisy trace that we've called V good, the ground truth parameters are recovered well as the mode of each posterior distribution is pretty much right on these black dashed lines. And the method has very little uncertainty about the inferred parameters, which is represented by the narrowness of the distributions. And then when we move to the analysis of this shorter, noisier trace, which we've called V poor, we see sort of how the method performs in a more challenging situation where the modes of these posteriors are near the expected values, but not quite on the mark. And the distributions are significantly broadened, showing that the method was definitely not quite certain of an exact value and found high probabilities for several, which is probably reflective of the effect of a larger noise level and a shorter trace length. So another important takeaway of this method is it allows a full characterization of the uncertainty, not only through these posteriors, but also through pairwise correlations of all parameters. So here I'm showing these correlations only for the poor case, where again, the breadth of these plots indicates the confidence of the method, and a skewness from the skew from a vertical or horizontal axis indicates the degree of correlation. And so now when we look at this, we can see a significant correlation between the decay rate and the width and the modulation amplitude, and the decay rate and the modulation amplitude. And so this is just indicative of how fully we can characterize the uncertainty with this method. But this is a lot of information in one graphic, and this is only for a single Gaussian case, so it just gets significantly more complicated with more complicated models. So for ease of interpretation, we condensed these correlation matrices or plots into a correlation matrix where across the diagonal it's showing the same thing, just inverted, and the intensity of the grayscale is respective of the intensity of the correlation. And so now it's clear very quickly these correlations between K and W and lambda and W and so on. And then we can focus on actually looking at the correlation plots for the parameters of interest, which is generally going to be the peak position and the peak width. So these simplifications become much more convenient in more complex cases. And so while the parameter posteriors most directly show what actually comes out of the Bayesian analysis, it's not generally what we care about the most. So what we're going to care about the most is these distance distributions, which any structural conclusions will be based, and the model fit to the time domain. So to show these quantities, we use what we're calling an ensemble of posterior predictions. And what this means is we draw a small set of random parameter vectors, somewhere between 30 and 100 from all of the pooled MCMC chain samples, calculate the associated distance distributions and noise-free time domain signals. So the time domain signals for both cases are shown here on the left with relatively good fit to the Deere data. In the middle, we show the residuals, so the fit between, the difference in the fit between the model and the raw data, where we're looking for a systematic deviation around this zero line. So not surprising that there's not much deviation in the good case and a little bit more in the poor case. And then finally, the ensemble of distance distributions here on the right. And so again, for the good case, there is really little scatter around this ground truth, and that's not surprising because the Bayesian analysis recovered the parameters with such little uncertainty. However, for the poor case, there's a significant spread of positions and widths, which means that we really couldn't be confident about the position or width from this data alone. And so we really want to emphasize that it's essential to look at this full ensemble of distributions rather than putting too much stock in one single trace. Even if we were to take the trace that's the maximum of the posterior, it would look relatively like this p-true value. it can be misleading because it discards all of the information about uncertainty that we've gotten out of this method. So what we want to be looking at is something like this, where we're taking the whole ensemble as a whole and not putting too much faith in any one distribution. So now most distance distributions of proteins are going to be asymmetric and multimodal, which means they're not going to be fit well by a single Gaussian. So we need to move to some more complicated models. Taking this relatively noisy Deer trace that was generated from this distance distribution, this bimodal distance distribution with one mode significantly weaker than the other. And since we know the ground truth, we can decide, okay, a 2 Gaussian model is probably most appropriate for this. So again, we show a subset of the posterior samples generated from the MC-MC chains. The model does quite well at fitting the data. We see very little deviation in the residuals. And finally, that leads to very little deviation in the scatter of the distance distributions, with maybe a little bit more uncertainty of this, this weaker mode. But now this is again sort of a pretty good case to look at. We want to look at a more challenging case. So we chose this broad distribution with several poorly resolved modes of similar intensities. And again, since we're fortunate enough to know the ground truth, we chose a 3 Gaussian model as the most appropriate. And although the model fits the data well, again, there's relatively little scatter around this 0 line. We see much more scatter in the distance distribution. And so what this is showing us is really, there's pretty much uncertainty about the shape over the entire range of this distribution. There's perhaps a little bit of certainty in the long distance edge, but the short distance modes, there's the possibility, it's showing the possibility of several of these modes, but nothing concrete. So again, this highlights 2 things: that we want to be taking the entire ensemble into account and not putting too much faith in any one distribution, and that even though we can have these really nice time domain fits with arguably very little difference, we can have a significant difference in the distance distributions that are output. So finally, everything up to now has been synthetic data, and that's nice because you can have a ground truth, and from that you can know generally what number of Gaussians is going to be most appropriate. But when we're working with experimental data, we're not quite so fortunate. So we need to talk about checking whether a multi-Gaussian model fits the data and comparing the quality of models across multi-Gaussians. So this is experimental data collected by a postdoc in our lab, Eric Evans, to measure the inter-subunit distance of a gated ion channel. And so we analyzed this Deer trace with anywhere from 1 to 4 Gaussians. And beginning with the first Gaussian, we see quite obvious deviation among these residuals, which is interesting because the distributions that are output are quite certain for this single Gaussian case. But even from what we know about the system, we could eliminate this as an appropriate model choice, since this distribution is believed to be at least bimodal. So then moving to the 2 Gaussian case, there is less deviation, but still some. And again, we see the distribution taking a bit more shape with a relative certainty of the positions. And then when we move to 3 and 4 Gaussian, there's little difference to be seen in the time domain fits, in the residual analysis, and the distance distribution. So it's possible that there is some overfitting occurring in this 4 Gaussian case, as it's not quite sure what to do with some of these longer distance modes. But just looking at this residual analysis would not be sufficient to tell me which of these 2 models is the most appropriate choice. So we need to combine this residual analysis with a tool that's already built into the Bayesian framework, which is what's known as a Bayes factor. And this Bayes factor, in a really simple explanation, is just comparing a ratio of the likelihood function of one model, say a 2 Gaussian model, with that likelihood of another model, perhaps a 1 Gaussian model. And so this really lets the data speak for one model over another. And so then on the right here, we've shown the log Bayes factors of all 4 cases shown on the last slide, compared to the single Gaussian case. And as these Bayes factors increase, that would suggest that each model is a better fit than the last. Though it's important to note again that there's very little difference between the 3 and 4 Gaussian case. So if I were just to look at the Bayes factors and then decide the 4 Gaussian is probably the best, that would likely be not the best choice because we've seen on the last slide that it's likely overfitting. So a combination of this residual analysis, the Bayes factors, and we're fortunate to get so much uncertainty quantification out of this method that we can also look at the correlation plots. So when we look at the correlation plot for the 3 Gaussian case, we see very little breadth in these modes, showing that the method was quite certain, especially of the earlier peaks and a little bit less so of the later distance peaks. And now when we look at the 4 Gaussian case, it's very indicative of overfitting. We have much broader range of data, which means the method really wasn't sure what to do with some of those longer Gaussian, or those longer distance peaks. So that all being said, combining all of this, we would probably say that the 3 Gaussian is the most appropriate choice, and this just highlights that it can be fairly difficult with an experimental case to choose the appropriate model, and it's essential to compare the different models with one another before choosing. So finally, what can we conclude from everything? This method really fully quantifies the uncertainty in the parameters from the raw deer data. Beyond that, it also allows us the ability to extrapolate correlation between the parameters, and then finally visualize these ensemble of distance distributions rather than just error bands. And for the future, we'll be looking to apply this Bayesian method to non-parametric models. So thank you so much to everyone for listening. And then I want to thank the entire Stoll Patrol for just being the best group, particularly the Stephans for being a fantastic team to work with on this Bayesian project. For those of you that know him, Thomas Edwards is the one who really got the group rolling on the Bayesian path in the first place. And just a quick plug, the Stoll Patrol has recently joined Twitter, so if you haven't followed us, give us a follow. Thank you again, and I'll take any questions.
Thanks very much. So if you have questions, the best way to do this is if you go to the participants panel, you can raise your hand virtually, and then I see you, I can unmute you, and then you're free. So, any questions? Dennis? I think.
Yeah. Hi, Sarah. So this was a really cool talk, especially because it was really helpful for me to understand some of stuff of the uncertainties. And my first question would be rather easy. So, or simple, not easy, I guess. Possible to estimate a plus-minus value for a specific parameter. So to say the position of the Gaussian is at this position plus minus half a nanometer from your analysis.
Yeah, I would say yes, because if you look at the posteriors themselves, so I can't go back.
Yeah, but that wouldn't
look
take.
At the posteriors themselves, it shows really like on a nanometer scale what the method is predicting is that parameter value, and so the mode is what you would generally take is the value, but then you could look at the entire spread of the distribution and say plus or minus so much.
So then you would take something like full width at half maximum or something like that. Okay.
Okay, thanks. Any other questions? Gunnar?
Okay, so you say it's an advantage that you have an ensemble information on the distance distribution rather than error bands. What exactly can you do better with the ensemble information than with the error bands?
So with the error bands, there's a few things that come up. You're assuming that it's a symmetric Gaussian distribution, which is not always the case. You're not capturing the correlations between the P at different distances. You're not showing the correlations between R naught and width. And so some of these can be avoided, like with Lewis's bootstrapping method, but there's others that can't. So when you actually show the entire ensemble, you're seeing all of these correlations and uncertainty actually visualized rather than just assuming this one shape with the error band.
Okay. More questions? Songi, I unmute you.
Hi, can you hear me?
Yes.
Okay, because I'm using my phone, I'm not entirely sure. So recently we have been working with polymers, and then if you look at a distribution of end-to-end distribution by its principle, the shape will be non-Gaussian because it's just the way end-to-end distance distributions work. So how do you solve such problems when the distribution is by its nature non-Gaussian?
Yeah, again, you would have to test with several models and try to see which is the best fit, but a Gaussian method might not be the most appropriate. So the nice thing about this specific method is that it's really easy to change the basis function of which we're actually using. So you don't have to use a Gaussian mixture model. That's just what we chose to exemplify the work, but you could easily use a Rice distribution or something else. And then ultimately, we hope to apply this Bayesian method to non-parametric models, in which case you wouldn't need to assume any specific shape of the distribution.
Rob? I think you can unmute yourself.
Hi, it's actually Graham on behalf of Rob.
Ah, okay.
So very, very nice talk. I enjoyed it lots. Sorry, I may have missed it because I missed the very start of the talk, but you know, often a significant source of error in when you're trying to work out distributions is correction for the background. So can you comment on, you know, is this method suitable for estimating errors there?
Yeah, so this method is nice because it just takes the raw deer data and gets out all the uncertainty without having to make that background correction. So then you don't have to assume or look for error for a background correction because there is none.
Okay, so in the examples you've given there, they're all raw data effectively?
Yeah.
Okay, well, very nice.
Okay, more questions? Dennis has another one. Yeah.
Yeah, sorry, me again. So another question to your slide number 18, roughly. They showed the differences in the time domain. Have you any experiences when looking at the spectral domain, if they, so the dipolar spectrum, if you get any better resolution of differences there?
I'm sorry, can you repeat the question?
So you look now at differences of the fit in the time domain, and you could, taking your fitted background into account, also calculate the form factor, and from that, the dipolar spectrum.
Right.
Do you see the dipolar, in the dipolar spectrum better or bigger differences than shown here for the 3 and 4 Gaussian case?
So are you asking like if I applied like Tikhonov, or if I applied something where I had a background fit and then took that fit and compared it to the raw data?
No, not exactly. So in principle, here is also a background fit included. So in your parametric model, there's of course a background or parameters for the background.
Yeah, there is a background.
From that, you could extract the background and could extract a form factor. Not that is any helpful for your analysis, but just for visualization of the dipolar spectra. And my question is then more, do you think it is helpful to look at the dipolar spectra or not?
I don't think so, but I mean, I don't feel like it would show anything different, especially because in the time domain, the fits already look pretty strong in most cases. So I don't know that it would show much. Maybe if there was a situation where the fit was quite terrible, then I would try to look at that, but I don't think so.
Okay. Thank you.
Okay, any urgent questions? Otherwise, I would thank Sarah again on behalf of everybody. And then I would say we switch speakers, and then it probably starts in